Very warm welcome to all of you guys. So today we will going to talk about Enterotoxigenic E.coli. So without any wasting time, let's get started. So today here we will going to talk about two type of toxin: heat level toxin and heat stable toxin. Heat stable toxin can be divided into two parts. One is STA, another one is STB. Okay, in some literature it is also mentioned as ST1 and ST2. So let's start our discussion. So first of all, let's talk about some generalized information from enterotoxigenic E. coli. That is, it is transmitted by water and is associated with traveler's diarrhea. Okay, so why it is known as traveler's diarrhea? The simple thing is, when a person from developed country visits developing country, and in the developing country he or she somehow drink contaminated water, then he and she can develop enterotoxigenic E. coli infection, and that is why the name traveler's disease. Okay, although the actual cause is, it is so called because of the classical clinical presentation, which is traveling to Mexico. Okay, where the patient drinks the contaminated water. So traveler's diarrhea, also sometimes known as Montezuma's revenge. Okay, so this is some historical perspective. So here you can only remember that enterotoxigenic E. coli are also known as traveler's diarrhea, and it is also known as Montezuma's revenge. Okay, and what is Montezuma? Montezuma, for information, was an Aztec emperor back in the 16th century who used to rule Mexico. Okay, so let's now talk about the main thing, that is the mode of action of two toxin produced by ETEC. So first we will going to talk about heat level toxin, then heat stable toxin. So I have downloaded a image to explain you both the heat stable toxin and heat level toxin. So let's move to the image. So this is a very common one that available in the Google for the mode of action of ETEC. So first of all we will talk about heat level toxin, then heat stable toxin. So before going to talk about heat level toxin mode of action, I want to mention that the mode of action of heat level toxin is exactly ditto of the mode of action preferred by Vibrio cholerae toxin. Okay, similar, exactly similar. Okay, so please try to concentrate now. So if you can learn this, that means you have already learned about the Vibrio cholerae toxin. Okay, so let's start it. First thing is, ETEC have to colonize, right? Remember that this is the pathogenic mechanism of bacteria, that first it have to colonize in the target cell, then it will either produce toxin or either doing anything to infect it, right? So first of all, colonization by their factor antigen. Okay, once colonization is successful, it will start producing AB type toxin. Okay, that I am telling as heat level toxin. So what is the meaning of AB type toxin? So if you focus this picture, then I think you can understand that it consists of two subunit. One is A subunit, another one is B subunit. That is why the name AB type toxin. So B subunit means binding subunit and A subunit means catalytic subunit. Okay, B subunit are of higher molecular weight than that of A subunit. Okay, so this AB toxin will bind via a specific type of receptor, or this AB type toxin bind with a specific type of receptor present on the intestinal epithelial cell. So remember that the target of ETEC is intestinal epithelium cell. So this receptor is GM1 ganglioside. Okay, so although it is not mentioned here, that this GM1 ganglioside receptor is the receptor where the B subunit will bind. Okay, then it will internalized, right? It will internalized, and after that it will be, there will be a time when the A subunit gets separated from the B subunit, right? As you can see, right? Because the color for A subunit is that one and color for B subunit is that one. That means it is obvious that A subunit is getting separated, right? So A subunit will be now released to the cytoplasm. Okay. So I hope up to that part it is very clear to you what is going on. First of all, colonization will occur. From after that, it will produce heat labile toxin. This heat labile toxin is a exotoxin and it is a AB type toxin. It will bind with the GM1 ganglioside receptor present on the surface of the intestinal epithelial cell. Okay, and then it will internalized. Okay, then inside the cell, the A subunit will be separated from B subunit. Okay. Now, what this A subunit will do, before going to discuss about this, I want to told you that A subunit or catalytic subunit interferes with G protein signaling. Okay. So, first of all, before going to talk about how they interfere with G protein signaling, it is necessary to discuss what is how G protein signaling operates. Okay, then it will be easy for you to understand. So we will learn about G protein signaling with the help of this picture. So remember that G protein signaling will activates only when G alpha subunit dissociated from G beta and G gamma. Okay, and G alpha during this time will have GTP. Okay, as you can see, I have drawn here G alpha subunit in a dotted line. That simply signifies that it used to be there. Okay, and during this position it consists of a GDP. Okay, clear. So now when G alpha is having GTP, it is fully activated and it will in turn activates adenylate cyclase. Okay, clear. So what adenylate cyclase will do after being activated, it will convert all the ATP molecules that are present inside the cell into cyclic AMP. Okay, until it is activated, right? Because after certain time G alpha will be go back to its inactivated state, and in the inactivated state it will have GDP. Clear. So this is the normal situation. Now let's talk about what interference our heat labile toxin along with Vibrio cholerae toxin introduced. So what they will do, this A subunit will take a ADP ribose group from NAD. Okay, that means A subunit are taking ADP ribose group from NAD. Clear. Now let's see here. I have told you that G alpha is with GDP when it is in inactivated state. Okay, now when it is required, it will convert in, it will take GTP instead of GDP. GDP is getting lost, right? So now under such circumstances it is activated, right? And as it is activated, it is obvious that it will activate adenylate cyclase. Clear. Now under such circumstances, what this catalytic A subunit will do, the catalytic A subunit will attach ADP ribose group that it takes from NAD into G alpha subunit. Okay, and during that time, remember it, G alpha is having GTP. Okay, so what will be the consequences? Remember it, when G alpha with GTP are attached with ADP ribose group, G alpha become locked into its on state. Okay, on state means G alpha cannot go back to its inactivated state. Okay, until ADP ribose is still with itself. Okay, so what is the consequence? The consequences is, as G alpha is activated for a longer time, it is obvious that it will activate the adenylate cyclase for a longer time, right? So now, as adenylate cyclase is activated longer time, it will keep converting ATP into cyclic AMP. Okay. As a result, much amount of ATP will be converted into cyclic AMP. Okay. So what cyclic AMP will do? Cyclic AMP will in turn activates protein kinase A. Okay. And protein kinase A will be activated for a longer time because cyclic AMP is produced in a higher amount, right? So protein kinase A will in turn activate Cftr channel. Okay. So what is the Cftr channel? Cftr channel is a specific channel for chlorine. Okay. All the chlorine ion are entering and also removing from the cell via this channel. Okay. And as it is activated for a longer time, and remember this, that Cftr channel usually used for the exit of chlorine. Okay. So as it is activated for a longer time, much amount of Cl minus will be getting lost from the epithelial cell into the bowel lumen, our intestinal lumen, not bowel lumen, intestinal lumen, right? So that is what going on here, right? As GTP is G alpha GTP complex is activated for a larger time because of the presence of ADP ribose group, it activates the adenylate cyclase for a larger time, which although for a longer time. Okay. And which in turn activated protein kinase A for a longer time. Okay. And this protein kinase A activation in turn opens Cftr channel for a longer time. Okay. And which causes much amount of chlorine minus ion lost. Okay. And as it causes much amount of chlorine minus ion loss or chlorine ion loss, so under such circumstances, to compensate this situation, much amount of water also getting lost from the intestinal epithelium into the intestinal lumen. Not only from the intestinal epithelium but also the blood vessel which is present below. From the blood vessel also water is getting evacuated. Okay. So as a result, you can see that much amount of water and chlorine ions are lost from the intestinal epithelial cell, and that will facilitate the situation we called watery diarrhea. Okay, clear? So that is the idea. So let's recapitulate it. So ETEC first colonized there and it will produce AB type toxin. This AB type toxin gets internalized. There will be a situation when A subunit will be removed from the B subunit in the cytoplasm. In the cytoplasm, A subunit will take a ADP ribose from NAD and attach this ADP ribose group to the G alpha when G alpha is having GTP, not GDP. Okay, that means G alpha become locked into its GTP bound state, and as a result it activates much amount of adenylate cyclase. Okay, this adenylate cyclase will convert all the near about all the ATP which is present inside the cell into cyclic AMP. Okay, and this cyclic AMP will activate protein kinase A for a longer time, and this longer activation of protein kinase A ultimately opens Cftr channel for a longer time. Okay, and which causes the loss of much amount of chlorine ions. Okay, and along with that much amount of water as a compensation are also getting lost from the intestinal epithelium. Okay, not only that, the water from the blood vessels are also getting lost. Okay, and this the high loss of chlorine and water ultimately facilitates a situation called watery diarrhea. Okay, so that is what going on by heat level toxin. Okay, now let's talk about heat stable toxin mode of action. So heat stable toxin will bind with guanylyl cyclase. Here I want to mention one thing. Don't get confused that heat stable toxin will also follow the same mechanism of action that Vibrio cholerae does, because Vibrio cholerae have only one type of toxin who follow the mode of action of heat labile toxin, not heat stable toxin. Okay, so heat stable toxin will stimulate guanylyl cyclase. Here another point to be noticed, that heat labile toxin are involved in stimulating adenylyl cyclase, and here heat stable toxin are involved in stimulating guanylyl cyclase. And what will be the consequences? Yes, you are right. The GTP molecules present inside the cell will be converted into CGMP molecule. Okay, so there will be a higher concentration than usual of cyclic GMP. Okay, and this cyclic GMP will convert or will this cyclic GMP will activate protein kinase A for a longer time. Okay, and this activated protein kinase A will open the Cftr channel for a longer time. Okay, and as a result, same situation will occur. It will cause much amount of chlorine ion loss, and which will in turn causes to the loss of much amount of water. Okay, so that is the situation observed in case of STA toxin or ST1 toxin. Okay, and STB and ST2 toxin, in case of STB or ST2 toxin, the mode of action is not known until now. Okay, so that is the idea of how the toxin produced by ETEC facilitates watery diarrhea. First one is heat labile toxin, which is involved in activating the adenylyl cyclase with the help of a process called ADP ribosylation. And the heat stable toxin, that is the first type of heat stable toxin, will activate guanylyl cyclase. Okay, which will convert much amount of GTP into cyclic GMP. Okay, and this cyclic GMP will keep activated protein kinase A for a longer time, and as a result it will in turn opens the Cftr channel for a longer time, which ultimately causes the loss of too much amount of water and chlorine minus ion. Okay, and that is how watery diarrhea is occurring via ETEC pathogen. Okay, so I hope you have understood the entire concept very well. So now I am simply showing the note that I have made for you, so you can take a screenshot so that you can prepare your notes with the help of this if you want. Okay, so here I have also mentioned about the heat stable toxin. So now let's talk about how you can diagnose ETEC, that whether your patient is infected with ETEC or not. So you have to collect the stool specimen, then you can culture them, or you can also prepare a broth culture. Okay, now with the help of the culture supernatant, you can perform biological tests, that is animal tissue culture. You can also perform some immunotesting such as radial immunoassay and enzyme linked immunosorbent assay. Okay, and you can also use some DNA gene probes for the detection of ETEC. So that is the lecture for Enterotoxigenic E.coli. So I hope this video will be helpful to you to understand the concept of Enterotoxigenic E.coli. So in the next day we will talk about another important type of E. coli, that is EHEC. Okay, so till then friends, bye bye.